Now that we've set up what the cross-bridge cycling is and how it starts, myosin head bound to actin. You can see right here, the myosin head is bound to actin. So we know then that means that they have a high level of calcium intracellular, so inside the muscles, which allow the troponin to go through a conformational change, shape change, and move myosin out of the way. So when we start here, this is the very beginning of that muscle movement. The cross bridge has formed the attachment phase. And that's what we finished off in the last video, the attachment phase. Next phase is your power stroke. In the power stroke, that myosin head is going to pivot. That power stroke, that pivot, is going to take this thin filament, which is full of actin, and move it in one direction, moving it towards the center of the sarcomere. So as the myosin heads work on both sides, this is the sarcomere. Myosin heads attach, pivot to the middle, attach, pivot to the middle, attach, pivot middle. The sarcomere is shortening because the thin filaments are being pulled towards the middle. That's the myosin head. Pivoting is the power stroke. But it's not just one movement. The reason this is called a cycle is we have to go through the power stroke and then restart the whole thing again, which means we have detachment. You must detach the myosin head. Now notice, ATP has come into the picture. That ATP is going to bind to the myosin head. Now, the binding allows for detachment to occur, basically breaking the cross bridge. So now we have the myosin head that's pivoted. It has ATP attached and detaches. We somehow have to get this myosin head back to its full energy state. That's referred to as cocking of the myosin head. That ATP will hydrolyze into ADP and phosphate. That's the PI there, phosphate. What this does is this hydrolysis is going to break ATP apart, releasing energy. That energy is used to reset the myosin head to its high energy state. Now it's ready to bind again and pivot. It's a cycle. So what we're doing here is, you can notice up top, we have the cross bridge formation. At that point, that cross bridge is set because of the high calcium levels. Troponin, tropomyosin move out of the way. Then you can see the pivot. That's the power stroke right there. The myosin head pivoted. It's moving the thin filaments. Then the ATP binds. That causes detachment. And then the ATP is hydrolyzed. The hydrolysis releases energy to reset the myosin head to its full energy state. Reset it by cocking it back to its full energy state. From there, it goes right back up and starts again. It's a constant cycle. Every contraction has all these myosin heads. Remember, they're pairs. Contract, pivots. Next one attaches, pivots. Attach, pivots. It keeps moving, almost like walking it down each way. And as those myosin heads keep working, the sarcomere gets shorter, the muscle overall contracts. Finally, we have moved the muscle. From the very beginning, we started with that neuromuscular junction. Then we moved into the idea of down the membrane, so action potential, propagation, or conduction. Going down the T-tubule, finishing off this excitation-contraction coupling, causing the movement of the troponin and tropomyosin by the high calcium levels in the cell, allowing the myosin head to bind, and starting this cycle right here. This is finally the movement of your muscle. So all this happens in a blink of an eye, very, very quickly. All of this nervous conduction into the muscle, and then the depolarization and movement of the muscle itself. So a lot of physiology, step by step of how we're going through these.